Hey, Amanda. Can you hear me?
Yeah, I can hear you.
You don't look so hot.
I don't feel so hot.
GI thing?
What'd you say?
Is it a GI thing you said?
Yeah, I think so. I'm not sure. It honestly might be my new medication that I'm on.
Okay. All right.
So, but I'm sure I will be doing better tomorrow.
Okay. 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 Yeah. There we go. Okay. Let's pray. Lord, thank you for this cold day. Please help Amanda feel better. Thank you for this time. Help us use it wisely in Jesus' name. Amen. All right, so we looked at the naming of nucleotides. This table from your book, it's table 8.1, shows you essentially a summary of the names of the different nucleosides and nucleotides. So adenosine is a nucleotide, the nucleoside. The nucleotide is adenolate. Adenosine, adenolate. We're going to see if it ends in no sine, the purine, it is a nucleoside. Whereas if it ends in anilate, or just nilate, then it's a nucleotide. So if you swap those two endings, it's either nucleotide or nucleoside. So adenosine, adenolate, then just deoxyadenosine, deoxyadenolate, guanosine, guanolate, deoxyguanoline, deoxyguanolate. Then it's similar for the pyrimidines, except pyrimidines don't end in sine, they end in dine, D-I-N-E. So cytidine becomes cytidylate. Deoxycytidine becomes deoxycytidylate. Thymidine is usually deoxythymidine. That is thymidylate or deoxythymidylate. And then uridine becomes uridylate. So you swap the dine for dylate. And that is the naming convention of the nucleotides. And we can go back and look at them and you can see the various structure again. The bases, if you can identify the bases and then determine if the sugar is ribose or deoxyribose, you know what it is. It's just that simple. So we look up here, this one here, that's adenine, ribose, so it's phosphate, so it's adenylate. Then this one here, that's guanine, so it has a phosphate. Without the phosphate, it's guanosine. With the phosphate, it's guanolate. And then over here, that's uracil. So uracil with the phosphate is uridylate. Without it, it's uridine. And then up there is cytosine. Without the phosphate, it's cytidine. With the phosphate, it's cytidylate. And then if we notice, what's the sugar here? The sugar?
Sorry.
It's 2-deoxyribose, 2-deoxyribose, 1, 2, 3, 4, 5 sugars. 1, 2, 3, 4, 5 sugars, 5 carbons in a sugar. Got a phosphate adenine, so it's deoxyadenosine. With the phosphate, it's deoxyadenylate. Over here, guanine. With the sugar, the nucleoside, it's guanosine. With the phosphate, it's guanolate, but this is deoxyguanosine, so it's deoxyguanolate. And then we have not uracil, but with the methyl group, the 5-methyl, it becomes thymine. So with the deoxyribose, it's deoxythymidine. And with the sugar, it's deoxythymidylate. And then up there, cytosine. With the deoxy sugar, it's deoxycytidine. But with the phosphate, it's deoxycytidylate. Deoxycytidylate. All right, so be able to name those on site. There are bases that are modified in DNA and especially in RNA. Now, the degree to which bases are modified in DNA varies from species to species. Many insects, for example, like the flies, the dipterans, very little modification. When you get up to the homopterans, the true bugs, they get a bit more, they've got much more modification. When you look at hymenopterans, bees and wasps, ants, not very much modification. So it's going to depend, fungi, very little, mammals, quite a bit. So it's going to vary from species to species. RNA, especially tRNAs, will have a lot of modified bases, same with ribosomal RNAs. Messenger RNAs will have modified RNAs, usually at the front of the RNA. Beyond that, messenger RNAs really aren't modified that much. So one of the most important modifications is the addition of a methyl group. And that addition of a methyl group is going to make the base bulkier. When you make a base bulkier, it changes the geometry a bit of the double helix. That change in the geometry can affect the way proteins interact with DNA. So there's a group of enzymes called DNA methyltransferases. DNA methyltransferases. This is a big group of enzymes, and when you take them and compare them sequence-wise, they fall into some discrete groups. So DNA methyltransferases, they're unfortunately separated into just 1, 2, 3, so on and so forth. So there's a 1, a 2, a 3, and then there's 3 A's and 3 B's and what have you. And the NMT1s, they seem to be responsible for so-called maintenance. So they're responsible for so-called maintenance DNA methylation. That means that when the DNA replication machinery replicates the DNA, if there's bases that are methylated, then the DNA methyltransferase 1 enzymes work during replication to make sure any bases that are methylated during replication, that the replicated strand ends up with compensatorily methylated bases. So it keeps methylation that's there. So these are your maintenance methyl transferases. Then the 3 group, the 3s, there's actually an A and a B and an L group. And these are the so-called de novo methyltransferases. De novo methyltransferases. New. Yeah. Yeah. So the A's and the B's, they seem to work during early development to establish new patterns of DNA methylation that were previously erased when the gametes were made. There are other for example, there's a 3S. The 3S doesn't seem to have any enzymatic activity. And then rats and mice have another version of 3B. It's called 3C. And it seems to be a version of 3C that just does other bits and pieces. And yeah, anyway, so these 3s, they're the de novo methylators. They make new patterns of DNA methylation. Sometimes those patterns can be involved in tissue-specific patterns of DNA methylation. The DNA methyltransferase 2s are a very odd bunch because they look exactly like a DNA methyltransferase, but these turn out that they methylate tRNAs. All things. So go figure.
So 3AB and L are all de novo.
These are de novo. Yeah, this one seems to work with these to increase their activity. So methylated bases in general are not that common. They're relatively rare, even in mammalian DNA. Mammalians use methylation more than other species, but it's a stark minority of the bases that are methylated. So methylated bases, don't get the idea they're super common, they're not. So here are some of the modifications you see in at least RNA, because with ribose, this will be with RNA. So the 5-methyl cytosine, that's pretty common in tRNAs and also in ribosomal RNAs. Then you get methylation of the adenosine. That's N6-methyl adenosine. This happens in the DNA of bacteria as well, but that's also seen in tRNAs, ribosomal RNAs, things like that. Then you get 5-hydroxymethyl cytidine. You see this actually in phage. For example, phage T4 will modify its bases like that so that it escapes host cell restriction mechanisms. And then the 2-N2-methyl guanosine, that makes guanosine quite bulky. By making it quite bulky, you now are going to change the structure. You don't see this in DNA. You pretty much only see that in RNA. Messenger RNAs are methylated at the cap. Usually the first and the second base, sometimes the third of the messenger RNA is methylated. Sometimes too the sugar gets methylated as well. That's the only part of the messenger RNA that's methylated usually. tRNAs have quite a few methylated bases. Keep in mind all of these modifications, they're made after transcription. So these are post-transcriptional. Same thing here. This is post-replication. So here are some of the modifications you see in tRNAs. These tend to be specific to tRNAs. So adenine is deaminated and is converted to inosine. Inosine is very useful in tRNAs because, as you can see, its opportunities for hydrogen bonding are quite vast. Then you have pseudo-uridine. Instead of being attached to the ribose here, it's attached over here. The enzyme that does this basically clips the bond, turns the uracil, and reattaches it. So that's called pseudo-uridine. Notice it's still uracil, it's just the attachment site that's different. Then you have 4-thiouridine, where the oxygen is swapped with a sulfur. Sulfur is quite a bit bulkier, and it's pretty electron demanding. Then you have 7-methyl guanosine. That's a pretty bulky base. When you pull this out, your chances of hydrogen bonding here are decreased. RNA methylation systems are very different from DNA methylation systems. Just keep that in mind. Even though one of these does methylate tRNA, but it's only a specific tRNA it methylates. It methylates the aspartate tRNA. Nucleotides also act as molecules for cell transduction, signal transduction. So they act as so-called second messengers. These are the cyclic nucleotides, cyclic AMP, cyclic GMP, and cyclic AMP is a abbreviation for adenosine 3' 5' cyclic monophosphate cyclic GMP. That's shorthand for guanosine 3' 5' cyclic monophosphate. I think you see why cyclic GMP is preferred Cyclic GMP is preferred. And the structure of these is really pretty simple. So the hydroxyl group here and the oxygen here, you essentially lose the water and you form a bond between the 5' phosphate and the 3' hydroxyl. Same thing with cyclic GMP. So adenylyl cyclase makes cyclic AMP from ATP. Guanylate cyclase makes cyclic GMP from GTP. This is very important in the vascular system. Well, so is this. But in blood vessels, smooth muscles, cyclic GMP promotes vasodilation. Nucleotides, when you link them together, they form phosphodiester bonds. Phosphodiester bonds. And this piece here, I think, is easier understood when I show it to you. So you have the phosphate here attaches from the 3' here to the 5' here, 3', 5', 3', 5', 3', 5'. You see that repeating theme over and over and over again, be it DNA or be it RNA. 3', 5', 3', 5', 3', 5', blah, blah, blah. Also, yes.
Sorry, is there something that runs 3 to 5? I feel like I remember that.
Nucleic acids.
Is it okay?
Yeah. So the 5' end is the front of the nucleic acid. So if you have a free phosphate attached to the 5' end, not attached to any base, that's the front. So for front, think 5. 2 F's. Front is 5. The tail has a hydroxyl group with no phosphate attached to it. So that's the 3' end, that's the tail. So an F and a T, 5 to 3. That's the back of the nucleic acid. Front 5' end and a 3' tail end. So these go from 5 to 3. They're made, in fact, from 5 to 3, as we'll see. So these molecules, not unlike proteins, have polarity. They have polarity. The phosphate is charged at physiologic pH. That's why it's called ribonucleic acid, deoxyribonucleic acid, because the phosphoric acid groups are ionized. That makes them acids. This means that the molecule will be very heavily negatively charged. That is a feature of all nucleic acids. They're quite negatively charged. And because of the phosphodiester bond, you have a polarity to the nucleic acids. So the front of the train is the 5' end. The caboose and the back is the 3' end. Front to back, back to front. The front will always have a phosphate because of the way it's made. The back will always be a hydroxyl group not attached to anything. So the 3' hydroxyl at the end, that's a 3' tail. 5' attached to phosphates, but those phosphates, when attached to anything, that's the front 5' end. Now you can write nucleic acids like this. This is a very convenient shorthand. We go phosphate, so that would be 5', 3 prime, 5 prime, 3 prime, 5 prime, 3 prime. 5 prime is up, 3 prime is down. 5 is higher than 3. With the phosphate in between it, put the base up here. So D, da da 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 da. Notice 3 prime N free hydroxyl group. 5 prime N free phosphate. RNA has a hydroxyl group at the 2 prime carbon of the ribose. Under basic conditions, that hydroxyl group is subject to base catalyzed deprotonation. If you deprotonate it, then that 2 prime hydroxyl group now becomes a very serious nucleophile, and it will attack the adjacent phosphate that cleaves the phosphodiester bond. Your book very nicely illustrates how this happens. Now, this doesn't happen with DNA. There's only a hydroxyl group here. But the base steals that hydrogen to make water, then this will attack here. It attacks here. You then get this cross-link thing that'll take up water and will resolve to something like that, but you've clipped this phosphodiester bond. Therefore, RNA is inherently less stable than DNA because it's subject to base catalyzed hydrolysis. So a short stretch of nucleotides. How short? The magic number is 20. 20. Short stretches constitute oligonucleotides. Longer stretches constitute polynucleotides.
So if it's 20 or lower, it's an oligonucleotide.
Yep. So are the bases actually bases when they're free? The answer is yes. They are not strong bases, but they are bases. They're subject to this tautomerization, where they go from the lactim to the lactam to the double lactam. Once you stick them in a polynucleic acid, this type of resonance decreases. Can still happen, but the rate at which it occurs is lower. But the bases, when they're free, absorb quite strongly at A260. A260. And A260 in the laboratory is a very good way to detect nucleic acids because of their absorption at 260. In terms of topology, the bases are nearly planar if you're purine and completely planar if you're pruning. And here you have the absorption spectra for the free bases, at least the nucleotides. You can see while it's not the maximum for all of them absorb pretty strongly at 260. So if you use 260, you will detect all 5 of these bases very effectively. So if you look at cytosine, its maximum is more like 274. If you look at thymidylate here, deoxythymidylate, it tends to peak just on the shy side of 270, more like about 268 or so. And over here, guanosine. Well, that peaks over here. That's roughly 250 or more like 247 or something like that. But if you hit 260, that will work for all of them. If you have stretches of nucleotides that you're using that are just one base, then you can use the one. Yeah, that's usually not the case. So I use 260 and it works quite well. Mendez, you're kidding everybody?
He's sleeping in the window.
He's sleeping in the window. Okay. All right, so the bases don't like water. They're planar. You wouldn't expect them to. Also, the ring structures mean that they can stack one on top of the other. Those stacking, hydrophobic stacking interactions can be collectively at least quite strong. And this is one of the things that's going to cause nucleic acids to have a hydrophobic core when they assume their structures in water. The backbone, of course, is quite water soluble, but the bases tend to not like water and they'll tend to associate with each other. They can hydrogen bond with each other. In DNA, of course, you have very specific rules of hydrogen bonding. Adenine always hydrogen bonds with thymine. Cytosine always bonds with guanine. And if you look at these pairing rules, you can see thymine and adenine up here. There's 2 sets of hydrogen bonds. And guanine and cytosine have 3. That makes them more thermostable. And I think you might look at this and say, that's the only way they can bond. It isn't. But when you hook the bases to the sugars and you put them in a polynucleotide and you want to make a double helix, this is the way they're going to hydrogen bond. Their means of hydrogen bonding, or excuse me, the manner in which they hydrogen bond is constrained when you put them in a polynucleotide, and then you make a double helix out of that. So these couplings, they constitute a so-called base pair, a base pair. These are the most energetically favored hydrogen bonds when they are in a polynucleotide chain and then interacting with another polynucleotide chain. And you can see these here. This means that your double helix is going to be extremely regular. It's going to have the same diameter, the same width, 20 angstroms from top to bottom. And it's going to twist. This twisting will be very regular. You'll have 10 bases per twist. There'll be 34 angstroms per twist, and the bases will each be 0.34 angstroms apart from each other. Very, very regular. And you can see this here. Now, this double helix only works if the polarity of each strand is reversed relative to the other. So this strand here goes from 5' to 3'. That means this strand here goes from 5' to 3'. That's the only way you get this regular structure. This arrangement, where they go in 2 different directions, is called anti-parallel. It's absolutely essential for the structure of DNA, which you see right here.
For some reason they went the same if they were parallel. Is it possible, maybe?
No,
No.
you wouldn't have a structure like this. You have a structure that's very irregular and therefore not nearly as stable. This molecule stores genetic information. Because it stores genetic information, you need it to be stable. You need it to be long-term stable. And only by having this very regular structure do you get this beautifully stable molecule that stores genetic information. This is the reason why you can have DNA in the fridge for years. And it lasts in a test tube, and it lasts a very long time. Early experiments show the DNA tended to fragment when you pipetted it. So people thought the DNA was very fragile. Turns out that most of their preps weren't very clean and they're full of nucleates. So large molecular weight DNA will be sheared if you pipette it through a very narrow barrel pipette at high speeds. But DNA in general is pretty durable. Pretty durable. Very good. Any questions about nucleotides before we look at how to make them? Amanda, any questions?
No, I'm good.
You're good? Okay. All right, then. We will soldier on. Yep. All right. There's two ways to make nucleotides. Salvage pathways, de novo pathways. De novo means you make them from scratch. Salvage pathways means you're going to use bases from the degradation of nucleic acids and you recycle them. Salvage pathway is clearly quite important. The de novo pathway is clearly used pretty heavily. And this pathway, I have to tell you, is the worst pathway you'll deal with in this class. Easily. Easily. This pathway is virtually identical in all organisms. It is a highly conserved pathway. And even though there are a couple of differences in some organisms, the way you make purines is virtually the same from bacteria to people. Now, purines are made one molecule at a time. So if we break down a purine, we can see that carbon, carbon, nitrogen, those come from glycine. Glycine. Let's use my highlighter. All right, so this one here, that comes from formate. This nitrogen and this one, they both come from glutamine. Glutamine. This also comes from formate. So two carbons, notice these carbons that sit between the nitrogens, they come from formate. This nitrogen comes from aspartate. And this one here comes from CO2. So this piece here comes from glycine. And that's essentially how you're going to make this puppy. Okay, ready? Take a deep breath. So this is from your book. All these pictures are from your book. And it starts with 5-phosphoribosyl 1-pyrophosphate. Now, you might look at that and say, how do we make 5-phosphoribosyl 1-pyrophosphate, PRPP? How do we make that? Well, that's a valid question. So why don't we ask ourselves, how do we make that? So let's go to the magic blackboard here. Okay, so PRPP. Okay. There. Okay. Okay, PRPP, this is 5-phospho ribosyl alpha pyrophosphate. So this gets made from ATP and also from just ribose 5-phosphate. So ATP and ribose 5-phosphate, that's going to essentially take the gamma and the beta phosphate of ATP, and it's going to transfer it to the number 1 carbon of this, and this enzyme is called ribose 5-phosphate pyrophosphokinase. Pyrophosphokinase. That's going to give you PRPP. So that's how you make PRPP, is through this reaction here. So you're going to burn an ATP right away just to get the starting material of this. And this then is going to undergo first reaction. It'll be transaminated, which I'll show you on the slide. Oh yeah, very expensive. That's why the salvage pathway gets used a good deal. Because if you can avoid spending this kind of energetic money, shall we say, you're going to do it. Okay. So the first reaction is a transamination. Notice that the pyrophosphate here is to activate this, because this reaction is not very energetically feasible. It is if you activate that. So you burned almost the equivalent of 2 ATP to make PRPP. Now that pyrophosphate just gets lopped off. Then this will tend to get hydrolyzed by an inorganic pyrophosphatase. That's going to pull the delta G further into the negative range. So this is glutamine, PRPP, aminotransferase, and it forms pyrophosphate beta D ribosylamine. So you have 5-phosphoribosyl 1-pyrophosphate. Now it's 5-phospho-D ribosyl amine. Ribosyl amine. Now this aminotransferase is a 135 kilodalton monomer. When it's a dimer, it's inactive. And the end products of this reaction scheme, inosine monophosphate, guanosine monophosphate, or adenosine monophosphate, binds to this allosterically and converts it into a dimer. And then PRPP, the substrate, will convert it back into the active monomer. So right away, we see the end products inhibit this first step. Inhibit this first step. And for those who are interested, although I doubt that's not many of you, in E. coli, this is encoded by the F gene. The F gene. Now, this ribosylamine is not very stable. So the next reaction tends to occur pretty quickly. And that's going to essentially, you're going to take glycine and you're going to attach glycine to this amino group here. And that's going to give you glycinamide ribonucleotide, otherwise known as GAR. And this is called GAR synthetase. GAR synthetase. So this will burn another ATP.
Sorry, a couple slides ago, you said PRPP converts. Did you say, is it? Active, okay.
Yeah. If this is in the dimeric state and you get more substrate, you'll convert this back to the monomeric state. And in the monomeric state, it's active. GAR synthetase. GAR is more stable than the 5-phosphoribosylamine. So this next reaction will occur quite quickly. Now you're going to use tetrahydrofolate. You'll use it twice in this reaction scheme, this pathway. This will be an N10 4-mil. So it's the aldehyde form. And this is GAR transformylase. GAR transformylase. And that will give you 4-mil glycinamide ribonucleotide, or FGAR. FGAR. And then FGAR is going to undergo another aminotransferase reaction. So see this ketone over here? See that one? That is ripe for transamination. Now remember, the nitrogens tend to come from glutamine. So that's what's going to happen. So you have FGAR aminotransferase. FGAR aminotransferase. And that will give you formal glycinamide ribonucleotide, or FGAM. FGAM. This enzyme is also sometimes called FGAM synthetase. FGAM synthetase. In humans, this enzyme is found on chromosome 17. Just a tidbit of worthless information there. Okay. Now, what do you see? Aldehyde, nitrogen. I see a reaction about to occur. And so you're going to cyclize this. You're going to cyclize this. And so you're going to draw out a water. In doing so, you'll also need an ATP. And that will make 5-amino imidazole ribonucleotide, or air. This is the F-gam cyclase, otherwise known as air synthetase. And you might remember that air is a side product from histidine biosynthesis in bacteria. But we don't make our own histidine, so that doesn't happen. But herein lies the connection between this particular pathway and the histidine biosynthetic pathway. Now, air, 5-amino imidazole ribonucleotide, is a precursor for the synthesis of thymine pyrophosphate in organisms that make their own thymine, which we don't, but bacteria do. Yep. All right. Now we've got a difference between bacteria and fungi and metazoans. So in the conversion of 5-amino imidazole to carboxy-amino imidazole ribonucleotide, we have a difference. So in humans, we have air carboxylase. We stick the carboxyl group here, just like that. So we get a carboxyl group there, and that's air carboxylase. Air carboxylase. Yeah, simpler.
It's just one step.
Sorry?
Is it just one step in humans then?
Yeah. In fungi and bacteria, you have the synthesis of N5-carboxylamino imidazole ribonucleotide, or NCAR. You stick the carboxyl group here. You burn an ATP. Then you just move it up to here with an NCAR mutase. So you NCAR synthetase, NCAR mutase in humans, other metazoans, you use air carboxylase. Air carboxylase. And in bacteria, you have these two enzymes, they form a complex. They hand these substrates to each other and everything, and it's pretty cool. Okay, so now we have carboxyamino imidazole ribonucleotide, otherwise known as CARE. So we go from air to CARE. From air to CARE. Now we're going to make CARE, we're going to make Sycar. And we're going to make this forbiddingly long named thing. Look at this. Have we seen that strategy before? Urea cycle. This enzyme has a lot of affinity in terms of structure, in terms of mechanism to the arginosuccinate synthetase. So notice how biochemically we're using a very similar strategy with an enzyme that has a lot of similarity. So 5-succinyl and succinyl 5-amino imidazole 4-carboxamide ribonucleotide, mercifully abbreviated to SICAR. Now, just based on what you remember from the urea cycle, what do you think is going to happen? Yeah, and you're going to take the nitrogen, and this will be released as fumarate. That's exactly what happens.
And does this come, if that brings back? the difference between, like,
No, this both happens in humans and
Okay. So
bacteria.
it's just that one step
Right.
before. and then separate.
So SICAR synthetase, SICAR lyase, this looks a lot like arginosuccinate lyase or arginosuccinase, as it's sometimes called. You liberate fumarate and you make the ACAR, 5-amino imidazole 4-carboxamide ribonucleotide. Boy, we're almost there, aren't we? We just need a carbon. Well, where are we going to get a carbon from?
Isn't there another thing there?
No,
No.
we need to make a ring out of this. What'll react nicely with the nitrogen? How do we do it over here? Right, aldehyde. We're just going to use it. We're going to use another aldehyde. So who carries aldehydes?
Tetrahydrofolate.
Tetrahydrofolate. So you're going to use N10-formyl tetrahydrofolate. You have ACAR transformylase. You add the aldehyde and you go from ACAR to FACAR. And formyl amino imidazole 4-carboxamide ribonucleotide. Take out of water, and Bob's your uncle. And there you are, inosine monophosphate. This is inosine monophosphate synthetase, takes it out, and IMP, inosine monophosphate, that's the end product of the general purine biosynthetic scheme.
Was the last enzyme was like before this? Was that 5
Transformylase, right. Transformylase. You see cases where some of these enzymes are bifunctional in some creatures and what have you, but they catalyze the same reactions, with that one exception, with the air carboxylase and so on and so forth. So IMP is the end product of the general purine biosynthetic scheme.
Can you go back one slide, please?
And then the synthesis of adenine and guanine, or adenylate and guanylate, is 2 steps. And once again, we're going to use our urea cycle example with two enzymes that look a lot like the urea cycle enzymes and also look a lot like these enzymes here, sycar synthetase, sycar lyase, so on and so forth. So we have adenylyl succinate synthetase, is GTP, aspartate, you make adenylyl succinate, then adenylyl succinate lyase, you liberate fumarate, and you have adenylate. And adenylate will feedback inhibit that enzyme right there. For guanylate, you're going to add a ketone here. So you use a NAC monophosphate dehydrogenase. You add water and you oxidize. That makes, this is xanthine. So this is xanthylate. And then you're going to use a xanthylate glutamine aminotransferase. So take amino from glutamine, transfer it here, you'll burn an ATP, make guanine, guanylate and feedback inhibits the step here. And the regulation is shown very nicely in this slide right here. All right. That is all for today. Monday, we have the exam, yes? Wednesday, thank you. And then Monday, we'll go over pyrimidine, which is much simpler. Much simpler.
And then that's all this really is, those two things, or does it continue with something else?
Then we go to the scavenge pathway.
Okay.
Then we go to nucleotide breakdown, which is also important. No, okay, Amanda, that's it for today. Get well, would you?
Thank you. Bye.
Bye.